Okay, good morning class. I'm Durgesh Mishra and once again we are here to discuss a topic over diffusion and osmosis. This is for class 9 and we are going to talk about two modes of transport which take place inside the cell. I had got a few doubts from you students. How does the components such as nutrients or the gases or other components, minerals, water, etc., which is required by the cells, reach the cell? So, I would like to tell you all these requirements are completed by the process of either diffusion or osmosis. So, today we are going to talk about these two topics, diffusion and osmosis. I have written the definitions over here, you can note it down. But before we get into the definitions of osmosis and diffusion, we need to talk about solution. We need to talk about what is a solution. The first thing which we need to talk about is what is a solution. And so let us see what is a solution first. So solution is something which contains a mixture of two or more components. Let us take an example. I am taking it here. So first of all, what is a solution? Solution is a mixture or combination of two or more components, generally in liquid medium. For example, salt plus water. When we dissolve salt into water, this becomes a solution. And in this we see there are two components, salt and water. So salt is called the solute and water is known as the solvent. So now what we have, solution is something which contains mixture of two or more things. For example, we have dissolved salt in water and this combination is called a solution. The salt is the solute and water is the solvent. So there can be many other examples of solution, such as sugar in water or any such thing. So how do we decide which component will be known as solute and which component will be known as the solvent? So this is very easy. The component which has been dissolved is the solute, and the component in which it has been dissolved is called a solvent. So salt has been dissolved in water. What has been dissolved? Salt has been dissolved. So salt is the solution, solute. And in what it has been dissolved? It has been dissolved in water. So it is a solvent. Sugar and water. What has been dissolved? Sugar. So sugar is a solute. And what has been dissolved? In which it has been dissolved? It has been dissolved in water. So water is the solvent. Generally, we call water as a universal solvent. That is, water is such a solvent, a component in which most of the things can get dissolved very easily. So, depending upon this, we talk about a new term which is called concentration of solution. So when we are mixing these things, suppose I am taking two glasses or two beakers, maybe I am taking two beakers, and in these two beakers I have taken 100 ml of water. In both the beakers we have taken 100 ml of water, and in this we have taken 1 gram of salt, and in this we have taken 2 gram of salt. So we see the amount of water in both the cases is same, but the amount of salt which has been dissolved is different. Okay, this means the amount of solute is varying, and when the amount of solute is varying, there comes a term called concentration. Concentration is defined as amount of solute dissolved in a solution. Amount of solute dissolved in a solvent. So, talking about sample 1, if we call this as sample 1 and this as sample 2, then the concentration of sample 1 is 1 gram of salt in 100 ml of water. Sample 2, 2 grams of salt in 100 ml of water. So this way we talk that we can say the sample 2 is higher in concentration as compared to sample 1. Sample 2 is higher in concentration as compared to sample 1 because amount of solute in this is higher. Since amount of solute taken in sample 2 is higher as compared to sample 1, that is why we can say that sample 2 is at higher concentration and sample 1 is at lower concentration. Because I am telling you this because we will be frequently talking about higher and lower concentration in this two definitions. So you need to understand what do we mean by higher concentration and what do we mean by lower concentration. Okay, so higher concentration means amount of solute is high, or lower concentration means amount of solute is low. There could yet be another case in which I can take multiple beakers, multiple samples and say I have dissolved 1 ml in 100 ml of water and then 5 ml in 150 and so on. I can keep changing the amount of solute and solvent both, and at such condition it becomes difficult for a student to understand which is at higher concentration and which is at lower concentration, because in that case the solute and solvent concentration both are varying. Here the solvent concentration has been kept constant, that is why it is very easy to understand which is at high concentration. But when the concentration of solute and solvent keeps on changing, what we need to do? We need to convert it into unitary method. In class 8 you must have studied unitary method. That is, will convert amount of solute in 1 ml of water or 1 ml of solvent, and this will help us to understand which sample is at higher concentration. Okay, now let us talk about types of solution. So there can be 3 types of solution. 3 types of solution. There can be 3 types of solution. The first is hypertonic, the second is hypotonic, and third is isotonic solution. There can be 3 types of solution. The first is hypertonic, hypotonic and isotonic solution. The first is hypertonic, the second is hypotonic, and the third is isotonic. It is very simple to understand the names hypertonic, hypotonic, isotonic solutions. There are 3 types of solution: hypertonic, hypotonic and isotonic, and these 3 have been decided on the basis of amount of solute in the solvent. This has been categorized as amount of solute in the solvent. If the amount of solute is higher, I am talking about solute. Okay, remember I am talking about solute while differentiating between the three types of solution. If the amount of solute is higher than solvent, is called hypertonic. If amount of solute is lower than the solvent, it is called hypotonic. And if amount of solute is equal to the amount of solvent, that is called isotonic. Simple. I have marked it. If amount of solute is higher, then hypertonic. If amount of solute is lower, it is hypotonic. And if it is equal to the solvent, it is isotonic. Hypertonic, hypotonic, isotonic. Hypertonic, let us consider this as, this is the solute and this is the solvent. Okay, solute is higher than solvent. If this is hypertonic. If it is equal, then isotonic. If it is lower, then hypotonic. Very simple. I guess you have understood the concept of solution. A solution is a mixture of two or more things. For example, salt and water. The component which has been dissolved is called a solid, and in which it has been dissolved is called the solvent. We have talked about concentration and we have talked about three types of solution. That is hypertonic, hypotonic and isotonic. Solution based on the difference in amount of solute. Now come to the main topic of today, that is diffusion and osmosis. Let us see the definition. Movement of solute particles from higher concentration to lower concentration is called diffusion. The movement of solute. And if here we see the movement of solvent molecules from higher concentration to lower concentration, from higher concentration to lower concentration through a semi-permeable membrane is called a osmosis. So, first let us focus on this one. Diffusion is the movement of solute molecules from higher concentration to lower concentration. The basic fundamental is that everything flows from high to low. Nothing flows from low to high in this practical world. Water cannot flow from lower level to higher level. Water has to flow from higher level to lower level. And this is, or I can say this implies to every condition. Every solvent or solution flows from higher concentration to lower concentration. And the purpose of this flow is to equalize the concentration. So let us see the first. Movement of solute particles from high concentration to the lower concentration is called diffusion. Okay, so take an example of perfume. If the perfume contains a solvent and the fragrance as the solute. Now, when I spray the perfume at the corner of my room, what happens is, in a fixed time, the particles of the solvent start to flow in the room. It will start to circulate in the room continuously. And a movement will come and a time will come when the smell of that perfume can be felt throughout the room. Why so? Because see, if consider this is the perfume bottle, I am standing over here, I press the bottle, the perfume got spread out. Spray came in this area. So what is the thing? The solute has been spread out at this area. What happened is the concentration of perfume has increased, because in the whole room there was no perfume, but as soon as I spread this, amount of perfume has increased in the vicinity, this area. So we can say that this area has higher concentration of perfume as compared to the rest of the room. And what will happen? The perfume particles will start to move in from higher concentration to the lower concentration. And after some time, the perfume can be smelled or felt its presence throughout the room. This process is called diffusion. Okay? Then we come up to osmosis. Osmosis is much like diffusion, but where we see the line is both the lines definitions come similar up to this point, but osmosis involves a presence of semi-permeable membrane. While teaching cell wall, I told you that cell wall or plasma membrane is semi-permeable in nature. In some books, the word semi-permeable can we see it is written selectively permeable. So, when there is presence of some biological membrane between the two, it's called semi-permeable membrane, and if something passes through that membrane, it is called osmosis. For example, we take in a dish, we take a raisin, kishmish, and fill it with water. What happens? What happens is the water starts to flow inside the raisin. You must have seen, if you take a black grams chana at your home and soak it into water, what happens? You'll see the water after some time. If you soak it at night, in the morning you'll see the water molecules have moved inside the gram. Okay, that is called osmosis. The outer layer of the gram has permitted the flow of it. In kishmish also you see the water flows inside. If you dissolve that with a little amount of salt or something, nothing is going to come out of the kishmish or the gram, but the water is going to move inside it. This process is called osmosis. So, now, I guess you have understood. So, the first point of difference in this condition in these two is number, I'll show you where the difference lies in this two. The first difference is, in this case, the movement takes place of solute, and in this case, the movement takes place of solvent. Second, semi-permeable membrane is present in this case. In this case, no semi-permeable membrane is required. No semi-permeable membrane is required. And the third point is, this process can take place in air. I mean to say this can take place in all solid, liquid and gas. Okay, but this takes place only in liquids. This process takes place only in liquids. This process can take place in any type of solution. This can take place in solids, liquid or gases. I think there would be a confusion about solids. How does a solute can move in solid state? So you can see this whiteboard for the example of movement in solid. How? You see. Solid, I have written a few things using my marker pen over this whiteboard. If I leave this, if I wipe this out just now, suppose I have written this and I am going to wipe it out with my duster. It will be wiped out very easily. But if you see it very deeply, you will find it has left some impressions. This is because this marker pen has got The ink has got diffused into the solid surface. If you leave this written material for over some time, or suppose a week, it will be tough to wipe it out simply by rubbing the duster, because the ink has got diffused over the surface. And this is an example of diffusion in solids. Okay, now example of diffusion in liquids. Though see, diffusion is taking place in liquids also, and it is taking place in gases also. So take the difference. Suppose I have taken 1 glass of normal water, and I have taken 1 glass of water with salt, and I have taken 1 glass of water with lemon juice. Okay, both is present separately. 1 glass with salt and other with lemon juice. And now I pour these two in the third glass. I pour this two in the third glass. You can then feel the presence of salt and lemon at every point in the third glass. When you drink that juice, the third resultant one, you can feel the presence of salt and lemon at all the times. Suppose I poured the lemon one first and then the salt one. It's not that when you start drinking that you'll feel the presence of salt and then will come the presence of lemon. Both the lemon and salt presence will come at the same time. This is diffusion in liquids. But when we talk about osmosis, this is not an example of osmosis, because there was no presence of semi-permeable membrane. If there is a presence of semi-permeable membrane, as the case of resins and ramps, that would be called osmosis. And diffusion in gases, you took the example of the perfume. Okay, so now onwards, when we talk about movement of some particle in and out of the system in a cell, you need to be very careful to observe that the process which is taking place is diffusion or osmosis, and that can be done on the basis of the lecture today. You need to see the definitions, you need to see the types of solution, the concentration, etc. Hope you got it. In the next class, we'll come up with the next topic, cytoplasm and the nucleus. Till then, stay safe. Thank you.